Hi, I'm Edurne Urrutia, and in this poster session, I'm going to talk about characterizing sex differences in rheumatoid arthritis, a systematic review and meta-analysis of transcriptomic studies. Previous studies have described sex-based differences in the epidemiological and clinical features of autoimmune diseases, including rheumatoid arthritis. In this regard, it is known that the immune system acts differently between men and women, and that sex hormones play a key role in such disparities. However, the differences in the molecular mechanisms are not fully understood. That's why a better understanding of these mechanisms could provide greater diagnostic precision and personalized treatments by sex. Therefore, the aim of this study was to identify and understand the molecular mechanisms underlying sex-based differences in rheumatoid arthritis. So, to do this, we identified the differentially expressed genes between ill women and men, according to the comparison shown in the poster. As we can see in the flowchart below, the goal was achieved in 3 steps. First, we conducted a systematic review of rheumatoid arthritis studies, including healthy individuals and patients, with gene expression data from either microarrays or RNA-seq platforms. For this, we checked the public repositories Gene Expression Omnibus, ArrayExpress, and ATEX. Second, we assessed the selected studies individually. This step included an exploratory analysis, a differential expression analysis, and a functional enrichment analysis of biological processes from the gene ontology and of KEGG pathways. Third and lastly, we performed a gene meta-analysis using a random effects model followed by a functional enrichment of the overall result. In addition, the results were synthesized using the Revigo tool and Gaussian terms. As a result, we retrieved 5 datasets with 570 samples from the Gene Expression Omnibus repository. Figure 1 displays a prisma diagram. It shows how the studies were filtered according to a standardized process, with a number of remaining datasets in each phase. After the meta-analysis, 10 genes, 90 biological processes, and 11 KEGG pathways were identified as significantly sex-dependent. The results were quite numerous, so they were summarized in 2 ways, using the Gaussian terms and the Revigo tool. Figure 2 illustrates the number of Go terms associated to each Gaussian term. And Figure 3 was obtained using the Revigo tool, and it's based on semantic similarity. Both figures suggest a higher activity of the immune system and a greater presence of energy precursors in female patients. Male patients instead showed an overrepresentation of terms related to protein biosynthesis and negative regulation of osteoblasts. In summary, the systematic review shows that sex is still an overlooked variable in biomedical research. Regarding the meta-analysis, women show a higher response of the immune system and a greater generation of energy precursor metabolites. The latter may be related to the energy expenditure required for the immune response. On the other hand, protein biosynthesis and negative regulation of osteoblasts were noteworthy in men. These mechanisms could explain some of the differences between the sexes. Thus, our findings could lead to the discovery of new therapeutic targets and be the basis for future research devoted to sex-specific precision medicine in rheumatoid arthritis. Thank you so much for your time and interest. See you soon.